So let's look at the DNA and RNA. We have already noted that the DNA and the RNA are basically the genetic material in living organisms. That means that they carry genetic instructions. These instructions are basically on what type of proteins or even RNA molecules should be made by the cells, because eventually these are the ones that determine the characteristics or the characteristics of living organisms, or basically the traits, what we call genetic traits. So in this session, we want to look at these two molecules in details and see what are they made up of. And of course, we start with a scenario. And that scenario is that we are having researchers who are studying the molecular composition of genetic material in two different organisms, the bacterium and the plant cell. Okay, what they do is that they extract nucleic acids and analyze their components using biochemical techniques. Their findings show that there are nitrogenous bases, that is adenine, guanine, cytosine, thiamine, and uracil in different proportions. Further, the researchers conduct an experiment to determine the stability of these nucleic acids at high temperature and their ability to direct protein synthesis in a controlled environment. So there are different research projects around this. So the questions for this scenario are: one is that based on the information provided, explain how the researchers can distinguish between the RNA and the DNA in their samples. And then the other one is, why is thiamine found in DNA but not RNA, whereas uracil is found in RNA but not in DNA? 3, the researchers noticed that one of the nucleic acids degrades rapidly at high temperatures. Which nucleic acid is that, and why? Why does it degrade fast? Okay. The other questions are: explain how the structure of the DNA allows it to be stable while RNA is more transient in nature. The fifth question is that the researchers introduce a radioactive label into the nucleic acids and observe the movements in the ribosomes. Explain how this helps in identifying the roles of the nucleic acids in protein synthesis. That if a mutation occurs in adenine, or let us say, if a mutation occurs where adenine is replaced by guanine in a segment of DNA, what possible effects could this have on protein synthesis? So the other questions are that genetic engineers in Uganda are working on modifying plant DNA to improve drought resistance. Based on your knowledge of DNA structure, what precautions should they take such that you don't have unintended mutations? And then lastly, suppose a new virus is discovered with RNA as its genetic material, predict how the replication might differ from that of DNA-based organisms. So let's go into this discussion. So we start with the DNA. The DNA is the genetic material in most living organisms. The DNA is a double-stranded molecule, meaning that it has two strands. And these strands appear like a ladder. So we say that the DNA is a double-stranded structure that has a ladder-like appearance. The basic units of the nucleic acids, because under nucleic acids we have the RNA and DNA. So the basic units are the same in RNA and DNA, and these basic units are the ones we call the nucleotides. So they are the monomers. They are the ones that are used to build the nucleic acids. Okay, so what happens is that you have repeating units of nucleotides, such that you have what you call a polynucleotide, which is basically a long chain, which is unbranched. Okay, so for the DNA, each of these nucleotides is composed of three components. That is the deoxyribose sugar, and then you have the phosphate group, and then the nitrogenous bases. And the nitrogenous bases are adenine, cytosine, guanine, and thymine in the DNA. Okay, so in our diagram, we have the phosphate group, that is one of the components. We have the sugar, which is a 5-carbon sugar. So deoxyribose sugar is a 5-carbon sugar, and then a nitrogenous base there. And the nitrogenous base can be either adenine, guanine, cytosine, or thymine. So the ladder-like structure that we have talked about is this. So you have one strand and then another strand. The way they run is that they run in opposite directions. Okay, and then in between them you have bonds, and these bonds are between the nitrogenous bases. These are actually hydrogen bonds. But if we just say that the DNA is basically like a ladder, it may not be absolutely true, because the ladder-like structure goes on twisting around the different strands, the two strands. So one strand moves over the other or around the other. So we have what we call a helical structure. So the DNA is a double helical molecule. Okay, so in between we have bonds, which are between the different nitrogenous bases. So the backbone of each strand is the sugar phosphate groups or components are the ones that form the backbone. And then it is the nitrogenous bases that project away. Okay, so this in between, you have this pairing. This is what we call the nitrogenous base pairing. Okay, so cytosine pairs with guanine and thymine pairs with adenine. That's what we call the base pairing rules, that you don't have any other base pairing apart from that. You cannot have cytosine pairing with, for example, thymine. It cannot happen. Okay, so the fact that one strand runs in the opposite direction with the other, so we say that is antiparallel. So that is antiparallelism in the DNA structure. So one follows this 5 prime, and then another follows the 3 prime. So the directions are different. Okay.